Can you see the slide? Hello?
No, we can't see it. Yes. Yes, this is visible. Okay, I'm seeing the slide. It's now visible.
Okay.
Now visible.
Anyway, we are going to talk about the Striga control methods. Now, if we are going to successfully control Striga, we need to understand the life cycle of this parasite so that we can come in with the interventions. For example, we may disrupt this germination process by using resistant genotypes or by coming in with the herbicides which can cause germination in the absence of the host. We may also disrupt this attachment of the striga on its host, probably by bringing in a genotype which may not allow this process to take place. We may also prevent germination by applying nitrogen and phosphorus to reduce the germination process. Or we may come in with the biological agents. And we can also prevent the flowering so that we reduce the striga seed bank. Now, it is important that we may understand the relationship of a host and the striga. For example, this relationship of sorghum grain yield and the total striga dry weight. Probably this graph is suggesting that we need very few striga plants to cause a serious reduction in grain yield. So when we see few plants in a field, we should not ignore them, otherwise they will cause serious damage. So these are some of the key points to note. Striga damages the crop before emergence. So this implies that if we are using hand hoeing, it can be extremely difficult to control the parasites. Also, a small amount of striga cannot be ignored because it can cause serious crop damage. Probably you may need to read this reference by Hen SJ 2009, control the striga conodrum. It talks about striga management in various crops and the problems associated with the controlling parasitic weeds. Then there's also this paper, Current Progress in Striga Management, which is fairly recent. You may also read about the difficulties in striga management. Now let us look at the host parasite interaction. Now, if a crop genotype is attacked by striga, if it is susceptible, it's going to support high striga emergence, and it should give very low grain yields. If the genotype is tolerant or gives tolerance, it can also support high striga emergence, but it can produce high grain yields. So if we have tolerance, we should come in and probably prevent the striga seed production. Otherwise, tolerance may promote a lot of striga seed banks. Even the susceptible varieties can also promote high striga seed banks because both support high striga emergence. But partial resistance, we have less striga emergence and slight grain yield reduction or no grain yield reduction. So if we get a genotype with the partial resistance, it could be the best. But should we get resistance or immunity, that would be ideal. That variety may not support any striga, and it can also produce high grain yield. Through breeding, this is what we expect to get. So if we are controlling striga, we need to have crop genotypes which can at least give us some yields. We now know that in sorghum, we have resistant sorghum cultivars. For example, SAR29, SAR33, SAR35, SAR37, and SAR16. These have actually been evaluated in this country and found to be resistant. Mutengwa in 2005 made crosses between SV1, which is currently recommended, and SAR29 with some resistance. He managed to identify recessive gene which control the low stimulant production and called it the LGS gene. So there is a potential of actually improving yield through breeding, especially in sorghums. Here there's a paper where we screened sorghum cultivars which showed some resistance to Striga asiatica. These experiments were done, or these evaluations were done using pot techniques under glass house and also field sites in Chundura communal area near Kwekwe, where there's a lot of striga. Now, there are also some resistant sorghum cultivars among the probably sorghum land races, known as Kasvikisire, Katandanzara, Mutode, Nyamudzi, Nterwan, and Igan. These were screened by Chanyoedza in 1996. They appeared to show some tolerance or resistance to Striga asiatica infection. Now, there's a paper by Guatidzo which has also demonstrated that there are some sorghum genotypes with the Striga resistance and also tolerance. Now, in maize, initially tolerant maize cultivars were identified. For example, CML 395, it's an inbred from Simit, and R201 hybrid appeared to tolerate maize cultivars. In fact, R201 appears to have some resistance. Then there are also some IITA maize cultivars with those names, which were screened in Tanzania, and they were found to to tolerate the presence of a striker. These were reported by Mbraga and Masawi in 2001. Now, the question which has been asked by scientists is that is there any striker resistance in maize? Because previously scientists were only targeting tolerance rather than resistance. So indeed, there are now some sources of striker resistance. These have been obtained from wild relatives of maize, which are basically two. The first one is Zea diploperennis. It's like a grass. It's actually a grass which can resist these parasites. We also have another grass known as Tripsacum dactyloides, which doesn't support striga infestation. So what happened is that at IITA, genes from Zea diploperennis were deployed to tropical maize germplasm through breeding. And a line was developed known as ZD05 with some resistance to Striga hemontheca, which is found in West Africa. So Zea diploperennis was crossed to tropical germplasm, and the hybrid reduced the Striga emergence. This then is Zea diploperennis, a perennial teosinde or wild maize. Well, it doesn't give us cobs as such. since it is a wild maize, but it has the sources of Striga resistance. This is Trypsacum dactyloides, which also possess genes for Striga resistance. So the line ZD05, which was derived from wild relatives of maize, can avoid infection, probably through its less branched root architecture. But more importantly, it can resist attachments due to Striga infection. It also doesn't support growth of the attached Striga, as was reported by Rich and Ijeta in 2008. Now, in Zimbabwe, there were also some screenings of maize, and there were some hybrids which appear to be tolerant to witchweed, although they were not resistant, but at least one could grow them in Striga infested areas and achieve good yields. Normally, when we are testing for resistance, we usually measure height because that is the very sensitive parameter to Striga infection. What you do is to compare heights of the plants which are infected versus plants which are not infected. If the differences in height are small, it means there's tolerance. If, like what is happening here, as opposed to what is there. So if a variety gives you a big gap in terms of differences, if they are significantly different, you know that this variety is very susceptible to Striga infection as opposed to this, which is tolerant. Of course, later you can measure grain yield, etc., but the height can be used as a screening tool for tolerance. Here you can see R201. You cannot differentiate between the two lines. One for the Striga infected maize and the one which is not infected, which means R201 is extremely tolerant to Striga infection. Look at the other varieties with the big gaps. These are highly susceptible. Of course, this one is also tolerant, but this one is susceptible. So we can use heights to distinguish or to separate those which are tolerant from those which are not tolerant, simply by measuring heights. Here there's a paper which you may also want to read, where new quality protein maize genotypes were found. Some of them were found to be resistant to Striga asiatica. You know, quality protein maize can solve some healthy problems in children, and it was being recommended that it should be grown in smallholder farming areas so that it can solve nutritional problems, uh, especially in children or even adults. So some experiments were done in Gokwe, but they were Striga was a serious issue there. But incidentally, some of these maize genotypes could tolerate Striga asiatica. So you may want to read this paper. There's also a paper which highlights the development of hybrids with resistance to Striga. In fact, some germplasm with ZD05 were actually obtained from IITA, and some lines were obtained from CIMIT, and at UZ some crosses were made by Professor Gasura. So indeed, there are now some hybrids which can actually resist these this Striga asiatic. I think for the first time we now have these hybrids. You can also read this paper, Breeding Strategy for Resistance to Striga asiatica, based on genetic diversity and the population structure of tropical maize lines. It's part of work which contributed to the development of maize with some Striga resistance. These hybrids are not yet in production, but later on they could be produced by UZ because it formed a seed company. So these could be some of the candidates which could be produced by the UZ University of Zimbabwe seed company. Again, this paper, Genome Wide Association Studies for Striga asiatica Resistance in Tropical Maize. This paper tried to identify genes which are responsible for resistance in maize. And actually, this paper is based on an MSc thesis done by Rwafa here at UZ. These are the pots or polythene bags which can be used during screening of maize for Striga resistance. You can see this is the maize which was cut. This is the parasite Striga asiatica. Again, you can see the polythene bags during screening where we have the striga plants which are flowering. And of course, these are some of the effects of striga on the maize plant. This is one of the experiments where maize hybrids, which were developed by UZ, were being evaluated in Gokwe communal area. So this area is striga endemic, but you can see the maize looks quite healthy in these striga infested areas. The maize was able to produce good cobs. These are the maize hybrids developed at UZ. You can see striga. This is in Gokwe during evaluation of the hybrids. You can see quite a big cob from the maize grown in striga infested areas. Actually, the farmers were almost abandoning these fields because of striga, where these maize hybrids were actually grown. Again, you could read another paper which is entitled Response of IITA Maize Inbred Lines, Bred for Striga Hemontheca Resistance to Striga asiatica and Associated Resistance Mechanisms in Southern Africa. So the materials which were acquired from IITA were later screened here in Zimbabwe at UZ. And you can read some of the results concerning the maize inbred lines. Actually, these maize inbred lines contained the ZD05, which is known for resisting striga. And these lines were later on used to develop hybrids suitable for Zimbabwe through a breeding program. Now we move on to tobacco tolerance to striga gesneroids, which is a problem to tobacco. In fact, the expansion of growing tobacco in Zimbabwe resulted in some of the tobacco being grown in striga gesneroids endemic areas, places like Mvuma, where striga is a serious problem. Now, after screening some of these tobacco genotypes, it was found that T66 was the most tolerant to striga gesneroids. This was followed by T61 and KRK26, according to Kogard 2011, who was working at the Tobacco Research Board. These are some of the symptoms or the effects of striga gesneroids impact on the tobacco. You know, on tobacco, the leaf is the one which is harvested, and the quality could be reduced by Striga gesneroids. Now, Koga, who was evaluating these genotypes, actually did his DPhil at UZ, entitled "The Genetic Characterization of Striga gesneroids and its Control Strategies in Tobacco." It's available on the UZ library. So he also wrote a paper, "Screening Tobacco Genotypes for Tolerance or Resistance to Striga gesneroids Infestation in Zimbabwe." He also found that Striga gesneroids was parasitizing a wild legume known as Dalbergia species and Ricardia scabra. Ricardia scabra is a weed. But I must say that after publishing this paper, a genetic analysis of the parasites found on Ricardia and Dalbergia were quite different to those found on tobacco. So probably, in fact, some literature assume that Striga gesneroids attacks Ricardia scabra and Dalbergia. Maybe it's not Striga gesneroids. Number two, mechanical methods can be used to control Striga, but this is problematic because it's not going to rescue the crop, but hand hoeing and pulling can prevent seed addition. And this can be done in February. This is when we see the parasite producing seeds. We may try to burn the striker. However, in the long term, we expect probably the situation may improve. But remember, striker seeds could stay in the soil for 20 years. So with these methods, we are expecting a very long-term mechanical control methods so that you see the impact. Herbicides, yes, there have been some herbicides which were used, like 2,4-D, MCPA, which were recommended, a long time ago in this country. Actually, these herbicides have been consistently been used on large-scale commercial farms, and it is thought that these herbicides could have reduced this problem on commercial farms. Since we now see Striga being associated with the smallholder agriculture. Please note that the damage will have occurred when these herbicides are applied. What we are saying is that even if when we use these herbicides, the parasite would already have caused some damage. And farmers may not want to invest in such kind of a technology which is not going to rescue their crop. They cannot just do it for management purposes. However, there's also another herbicide known as dicamba or banvel, which can be applied post-emergence around 4 weeks. This one can be translocated, meaning that it can move within the maize plant downwards and reach the roots, and it can kill the parasite. But the efficiency of dicamba depends on how it is being translocated to the roots, and this may differ across maize genotypes. Then we have imazapa resistant maize technology, or IARA maize. This was developed by CIMIT and has got two components. We have a herbicide resistant maize seed, which is IARA maize. The second component is the herbicide imazapa, which is a systemic imidazolinone. Now, what happens is that the herbicide-resistant maize seed is dressed or coated with about 30 grams imazapa per hectare to control Striga. So during the seed production of this hybrid, the herbicide is added as a powder. It's already within the seed. It's seed dressed. The farmer doesn't have to spray this herbicide. It's already on the seed. That is the smart. It looks like a smart technology. So you plant your maize having the herbicide with it, and when Striga tries to germinate after all the preconditioning processes and the presence of the stimulants, it is met with the imazapa, and it disrupts the germination process. Then the maize will survive. Now, there are some papers which you may read concerning this technology. Imazapa herbicide seed dressing increases yield, suppresses Striga asiatica, and a seed depletion role in maize in Malawi. This was published by Malawi scientists who tried this technology. The technology was also evaluated in Zimbabwe, but across many countries. So experiments were pulled from various countries in a multi-site, multi-season field tests demonstrate that herbicide seed coating, herbicide resistance, maize controls Striga species and increase yields in several African countries. So you had various scientists who did the same experiment and pulled their results together, including Zimbabwe, represented by Lawrence Jassy, who is one of the authors there. So you may read this and you may find the effectiveness of, you may find how the effectiveness of this imazapa was evaluated. In Zimbabwe, I think the sites were in Chundura communal area, which is between Gweru and Kwekwe. Crop rotations. There are some crops which produce germination stimulants and cause the parasite to germinate, but the parasite will fail to get attached to the trap crops. And this can actually reduce the Striga seed bank and makes it safe for maize and sorghum to grow. Now, among the bambara nuts, there are some bambara nut genotypes which were found to be very efficient in germinating Striga asiatica, and also among the cowpeas. In fact, there's this paper where the screenings were done, because it is important to find a variety which can produce a lot of germination stimulants for the control. I mean, for to allow to allow the germination of the parasite. Yet the parasite is not going to attack that genotype. But of course, in Zimbabwe, we do have two parasites uh, or more which exist in the fields, in the same fields. We may have Electra, attacking these crops. That is one of the disadvantage of using trap crops. Another one is known as catch cropping. A susceptible host is grown, and then you come in and plow it under. You destroy it. You destroy it with a plow. It can reduce the Striga seed bank. In Zimbabwe, Sudan grass was actually used for this purpose, and it can be done in abandoned lands. But this could be a problem with the farmers. Farmers may not want to grow a crop just before it produces seed, they destroy it just to kill Striga. This approach may not be feasible in terms of economics. So there's a drawback for using this kind of technology. Intercropping. This is when you can mix crops. For example, maize and cowpeas grown in the same row were found to reduce striga emergence, and it increased grain yield. This was reported by Kasembe Tar, 1998. Kasembe did an MPhil here at UZ on intercropping as a strategy to reduce Striga asiatica in maize. Khan et al. Also used intercropping. They intercropped maize with the Desmodium cynatum, which greatly reduced the Striga monthika in Kenya. Probably you may read a paper by Kasembe where he used intercropping as a strategy. Sorry, where he used intercropping as a strategy to reduce cowpeas. I mean, to reduce striga using cowpeas. This is one of this data is from Kasembe. You can see where there's sorrel maize, there's low yield. This is in striga infested areas. If you put cowpea, there was an increase in grain yield. It didn't matter which cowpeas, but of course there are some cowpeas which are better in terms of increasing the yields. This paper is by Khan, where they use the desmodium intercropping in what they call the push-pull technology or strategy. This is a climate smart push-pull. Let me just explain what it means. Now, here we have got sorghum intercropped with the desmodium. And we have got this grass, bracharia grass, which is on the edges of the field. So what happens is that this strategy controls two pests. Number one, it controls the striga, and number two, it can control stem borers. So desmodium will produce some chemicals which disrupts the life cycle of striga, and it will be safe for the sorghums. It will also produce some chemicals which will drive away the stem borers to this bracharia grass. The bracharia grass may also produce some chemicals to attract this pest. So desmodium will push the insect out of the field, and it will be pulled by the grass which is outside. That's why it is called maybe the push-pull strategy. And of course, it will take care of the striga, in addition to controlling stem borers. Again, here we have got the desmodium planted between maize. Previously, it was planted between sorghum plants. And on the edges of maize, we've got napier. So desmodium will disrupt the parasite from attacking maize, and at the same time, it will produce these chemicals which will push out the stem borers, and they will be attracted by the napier, and they won't cause any harm there. So the maize is free of maize stalk borer as well as the striga in this push pull technology, which doesn't involve, use of agrochemicals. But anyway, we are interested in the desmodia controlling striga in intercropping. So it's one of the technologies which is used. Now we move on to nitrogen. Nitrogen can also reduce the impact of striga in maize. Now, the effects of nitrogen depend on the crop cultivars. Agobli in 1991 used 120 kgs per hectare nitrogen to induce striga tolerance in maize. But 120 kgs per hectare is probably too much for the farmers. So farmers may need low rates to do this, to control striga or to induce tolerance in their crops. In Zimbabwe, we found out that CML 395 and R201 just required 30 kgs per hectare to achieve tolerance. And probably this could be affordable to farmers. In tobacco, about 50 kgs per hectare nitrogen could decrease the incidence of striga gesneroids in tobacco. Now, what would be the mechanisms of nitrogen effects on striga? Number one, nitrogen could decrease the number of germinating striga seeds. So, nitrogen can disrupt germination of striga seeds. The other point is that in the presence of nitrogen, there is low root exudate production. So we can limit the striga germination stimulants, which cause striga seed to germinate. So we can also reduce those germination stimulants. The other one is the inhibition of striga attachment process on the host roots, forming hostoria. So hostoria formation is disrupted in a nitrogen environment. The fourth impact is that nitrogen actually stimulates photosynthetic activity of the parasite and the host. And probably this can reduce the impact of the host, of the parasite on the host. Then high photosynthetic rates may also induce tolerance in the host. Now, phosphate was also found to limit production of strigalactones or germination stimulants, particularly sogomol and 5-deoxystrigol. These are just germination stimulants, and therefore it reduced striga hemontheca emergence. In this case, they used diammonium hydrogen phosphate. You may have to read the paper by Jamir in 2012. Now, an MSc student at UZ demonstrated that the addition of phosphorus could reduce the attachments on the attachments of Striga on a maize root. This is the maize roots with the Striga attachments. So if phosphorus was increased, it actually decreased these attachments up to very few. This technique is known as the agar gel technique, which is used to screen germplasm for resistance. But in this case, phosphorus was added to the petri dishes in order to evaluate the impact of phosphorus on the germination stimulants. We can see there were 6 maize genotypes, and as phosphorus was increased, you could see a decrease in all the varieties, decrease in the germination of Striga. So phosphorus can help to reduce Striga in maize. Timely planting. Now, early planted maize had high Striga infection, but it produced high grain yields. We thought that by planting early, maybe we could reduce the infestation, but it was opposite. But the good thing is that the maize tolerated the high infestation. So early planting should be recommended. But of course, it should be supported by hand weeding or pulling to avoid Striga seed addition. So farmers should be encouraged to plant early in Striga endemic areas. Now, in this report, Striga activity in Zimbabwe, we have got various experiments where time of planting was actually evaluated. And indeed, early planted maize gave high yields despite Striga infestation, although early planting could not control Striga. Biological control. Yes, there are opportunities for controlling Striga using biological agents. We have an insect known as Simincronix, which was found to control Striga montica in West Africa. Then we have also Fusarium isolates. This is a fungus, which could also be used to control Striga. Well, Simincronix and the Simincronix, probably it may not be applicable in Zimbabwe because we also need to check whether Simincronix may not be a potential pest. It has to be thoroughly checked. Then Fusarium, the same thing. But there are opportunities for probably developing a Fusarium as a strategy in controlling Striga in Zimbabwe. Then we have a vascular mycorrhiza, which was found to be effective by Ledzemo in 2007. Now, what may happen is that the cereal may provide fungus with carbon, because a vascular mycorrhiza is a fungus. Now, it can be provided with carbon in exchange of available phosphorus, because the vascular mycorrhiza are produced, occupy roots of crops, especially in low fertility, in poor fertility soils. Their function is that they can extract phosphorus from those soils and make it available to the plant in exchange of carbohydrates from that plant. So basically, the plant will benefit from the uptake of phosphorus. And the production of Striga lactones attract a vascular mycorrhiza to colonize the roots of the host. So we think that probably the high levels of phosphorus could actually have a potential in decreasing the parasite. And in any case, the Striga lactones for a vascular mycorrhiza and those for Striga are different. So if we breed crops for low Striga lactone production, we may not affect the requirements for the vascular mycorrhiza. And this has actually been demonstrated in some papers. So you may want to read this paper on a vascular mycorrhiza fungi, how it can be used to reduce the problem of Striga. This is a biological control. Again, another paper on a vascular mycorrhiza. Now, I just want to indicate that as far as electravogeli and Striga gesneroids are concerned, the life cycle is similar to that of Striga asiatica. And the control, probably hand hoeing and pulling could be done just to reduce the Striga seed bank. But what is interesting is that there's a cowpea known as B301, which was found to be resistant to both electravogeli and S. gesneroids, Striga gesneroids. So this has actually been used to develop some hybrids with resistance, and these can be screened and probably adapted in Zimbabwe. The other issue is that phosphorus applied at 45 kg per hectare was found to reduce the impact of electravogeli on cowpeas. We now know that phosphorus can reduce the production of Striga lactones. Probably this is the mechanism which explains why electravogeli is reduced. Now, there is also need to identify cowpea and bambara nut which are resistant or tolerant to these parasites. There's a paper on host-parasite relations between cowpea and Alectra vogeri, which you may read because the work was done in Zimbabwe. Also, the response of cowpea genotypes to witchweed Alectra vogeri infection. Now, the response of genotypes to witchweed Alectra vogeri infection were also evaluated by a BSc candidate using these polythene bags. You can see the cowpea, and these could be the effects on the leaves. It could be the presence of the parasite, Alectra vogeri. This is the Alectra vogeri in polythene bags, which is actually producing some flowers. Now on Cascuta campestris, we said it looks like a spaghetti on its host. If you see something like this, it could be Cascuta campestris. It is a problem in Lucerne. Also, it parasitizes tomatoes, carrots in the US. But in Marondera Horticultural Research Center, it has also been reported in tomatoes. Shoot parasite, it is a shoot parasite with little chlorophyll. It can be yellow to orange in color. It looks like a spaghetti mass on its host, just like I have indicated. Here, I just want to explain a bit on the life cycle. What you should remember is that this parasite doesn't require germination stimulants like Striga species. It only needs temperatures around 30 to 33 degrees Celsius plus moisture. If there's no dormancy, you get straight germination. Now, after germination, you get the plumule being elongated, and it will start to move in an anti-clockwise direction, which we call circumnutation. It will be looking for the host. Probably it is attracted through some chemicals in the air. We are not sure. Once it gets into contact with the host, it grows right around the target, right around the host branch. Now, there are some points of contact which forms the hostoria. And this hostoria enables the parasite to extract water and nutrients from the host. There's a substance known as cutin. It's like a glue which helps the parasite to get attached to the host. The host can also produce cytokinins, which are essential for this association. So you have got cutin, you have cytokinins, which are essential for the hostoria formation and forms a bridge between the host and the parasite. Then the parasite can benefit in extracting the nutrients from the host. After that, the parasite will form flowers. This could be white flowers. Then it forms seeds. The seeds can drop to the ground, and they have a hard seed coat, which enables them to have dormancy, and they can survive for 50 years. So it's quite a problematic parasite. So if I can just recap on the life cycle, we have got the germination, where we have hard seeds to ensure dormancy and can survive for 50 years. Temperatures between 15 to 39 Celsius, can allow germination. But no germination stimulant is essential for this process. When the seed is germinated, it forms a single shoot. The shoot swings in a circular anti-clockwise motion about once every hour. So during the day, you can do a lot of measurements on it. We call it circumnutation. It forms coils around the host, forms ostoria, cutiny, aids the bond between the parasite and the host. Now, ostoria development will require cytokinins, which are produced by the host. What are the effects of the parasite? It can reduce growth of the host. It depends on the host for water, mineral salts, and carbon. Photosynthesis in Cascuta is not efficient because it doesn't contain, it contains very little chlorophyll. Now let us look at the control. Because this is a shoot parasite, if you try to control it, you can also kill the host. That is the challenge. You can do hand pulling if you can see those seedlings. And you can prune on infected plants. You try to chop some of the infected plants to prevent further parasitism. You can burn and destroy the seeds. In Ruseni, mowing can be done to remove the parasite. Sometimes it helps. There's also need to identify Lucerne which is resistant and tolerant to Cascuta. That work needs to be done. Then herbicides, you can actually spray pendimethalin or stomp or trifluorine. These can interfere with the seedling at the seedling stage. So timing is very important. Or you could come in with a risky herbicide application where you can use gramoxone and glyphosate, which can potentially kill your host, but try to use dilute herbicides, hoping that if you kill the parasite, there's a chance that your host will recover. Normally, 75 to 150 grams of active ingredients are recommended, but calibration of herbicides will be something else. And there's this could be a very risky operation. Now, in conclusion, we would like to say that parasitic weeds are some of the most important weeds in Zimbabwe, and there is an urgent need to look for methods of controlling them. Use of tolerant or resistant genotypes should be the focal point of developing integrated control of Striga. And there is no single method which is likely to be useful to control the parasitic weeds. So there's a need to pool a lot of methods and develop integrated approach, probably using the resistant or tolerant genotype as the focal point, because you want to produce a grain yield from the crop. This then is the summary, which is a quick summary. Please note the parasitic weeds we have gone through. We have got root parasites, Striga asiatica, Striga foppsii, Striga gesneroides, Electra vogeli, Ramphicarpa fistulosa. Then shoot parasites, we have got Cascuta campestris, and this is the life cycle which is applicable to most of these parasitic weeds. You have to go through this life cycle. Then we have got the effects on the host. Wilting of plants when moisture is available, it may mean that you have got a parasite. Standard plants reduce the heights, reduce the stem biomass. These are the effects of the parasite. Reduced photosynthesis, photosynthetic rates in the host leaves. Accumulation of abscisic acid ABA in the leaves, leading to reduced stomatal conductance. Hormonal disturbance, ABA, gibberellic acid, and cytokinins, resulting in the alteration of plant architecture. Movement of carbohydrates to the root, where you have higher root shoot ratios in infected plants. There could also be production of toxins. I put a question mark there because they are yet to be identified. Then we have just gone through the control methods. We talked about first, tolerant crops or resistant crops, hand weeding, herbicides, 2,4-D, MCPA, and dicamba. Then this technology where we have imazapa plus imazapa resistant maize. Crop rotation, catch cropping, intercropping, soil fertility management, use of nitrogen and phosphorus in the soil. And then biological control, use of Synchronix, Fusarium isolates, and vascular mycorrhiza. So this is it on the parasitic weeds. We have to stop here.